Last but not least, in Chapter 2 is nucleic acids. This one that you're looking at right here happens to be deoxyribonucleic acid, or DNA. So there are two kinds, DNA, which I just said, and RNA, which is ribonucleic acid, and we'll go into the structural differences between the two in a couple of minutes. The important thing to know about these polymers is that the monomer is a nucleotide. This is a nucleotide over here. You've got a phosphate, a sugar, plus a base. You're going to see later on there are 4 different kinds of bases, which makes for 4 different kinds of nucleotides. Both your RNA and DNA contain information. They code for amino acids that are going into proteins, and the information is contained within the bases. So it's the sequence in which the nucleotides are put together with their representative bases that makes up the information, or that contains the information. And then all the bases are held in the right order by what we call a sugar-phosphate backbone. So here's DNA again. This is one nucleotide, and it's sugar, and then dark purple is the phosphate, light purple is the sugar on the next nucleotide, with its base sticking out in the middle. So you've got sugar, phosphate, sugar, phosphate, sugar, phosphate. That's your backbone. That's this strand right here is holding all of those bases or nucleotides together in the proper order. As we said, there are 4 different kinds. You don't need to be able to list or write the words, just know the abbreviations A, G, C, and T. And then each molecule of DNA contains 2 strands. So all of the nucleotides within a strand are held together by the sugar-phosphate backbone. But when the 2 strands come together, what keeps them from falling apart is the hydrogen bonds. So again, if you look, you've got your sugar-phosphate backbone on one strand and the sugar-phosphate backbone on the other strand, with their respective bases sticking in towards the middle, and the bases like to form hydrogen bonds with each other. So that's a DNA molecule, or a chromosome really, is basically 2 really big long molecules that have been velcroed together with these hydrogen bonds. If you want to look a little closer at how it works, here you've got one nucleotide. If you want to make a strand, you join to it other nucleotides. So you've got the sugar from this nucleotide is joined to the phosphate of the nucleotide below it, and then that sugar is joined to that phosphate. So these are covalent bonds. So you've got alternating sugars and phosphate, sugar and phosphate, held together by covalent bonds, forming one strand with the different bases sticking out towards the middle. On the opposite strand, you will always find what's referred to as the complementary base, or the complementary nucleotide. So anytime you see an A or an adenine on one strand of the DNA, across from it, hydrogen bound to it, will be a T. Anytime you see a G, there will be a C on the other side. So to make the complete the complete DNA structure, you take 2 strands, strand 1 here, which we already saw, and strand 2 here, and you stick them together, and they're held together by these hydrogen bonds. So here we just have AT, AT, TA, but in a long strand there would be G's and C's in there. And one of the things I'll point out right now, which isn't so important yet, but it will be later on, is there's directionality to the 2 strands. If you look at this strand right here, it ends at a phosphate, and the oxygen is pointing up. Over here, this strand has the oxygen pointing down, and it just ends at the sugar. So when you pull apart 2 strands of DNA, like this strand and that strand, the very ends of them are different. So the 2 strands have directionality to them. So that is DNA. DNA codes for proteins, and it always stays in the nucleus. Your cells guard it very carefully. When it comes time to make a new protein, and you need to get the information that's into the nucleus out to the cytoplasm, that is where ribonucleic acid steps in. So it is going to have 3 of the same nucleotides, A, G, and C, only in the case of RNA, instead of thymine as the 4th nucleotide, you have uracil. The sugar phosphate backbone is the same, with the exception that it's a different sugar. The big difference between the two is that RNA is single-stranded. And what RNA does is serve, as it says here, as a disposable copy of the information that's stored in the DNA. So after an RNA is shipped out to the cytoplasm and used to make a protein, it can be degraded and thrown away. So this is what the RNA looks like. You've got sugar, phosphate, sugar, phosphate, sugar, phosphate, with the bases sticking out, only there isn't a complementary strand on the RNA. It's just made one strand at a time. We will talk briefly about adenosine triphosphate because it is a form of nucleotide. This particular form isn't what you see in DNA. The adenine nucleotide in DNA has 1 phosphate on it. But adenosine triphosphate is your body's favorite energy molecule. So what your cells do is burn glucose, and then the energy from glucose is used to form ATP. And ATP is the molecule that drives all of your cellular processes. These processes include things like moving charged molecules across the membrane, pumps that would pump sodium out of a cell or potassium into a cell. The energy is used for physical processes like muscle contraction, and then also to promote chemical work. If you need to run chemical reactions that require energy, like building stuff up, it's ATP that gives that energy. That's it. That's video 3, and you're done with chapter 2.